Next, we also have the investigation section in this chapter. According to specification number 3, we need to investigate and describe the effect of temperature on respiration in yeast. So, let's take a look at this practical activity in detail. And, before beginning any practical work, it is essential to follow certain precautions to ensure accurate and reliable results. First, the same concentration of yeast, glucose, and methylene blue should be used in all test tubes to maintain consistency. The pH must be kept constant to prevent enzyme denaturation, which could affect the outcome of the experiment. Tubes should not be shaken excessively, as turbulence can alter oxygen availability and interfere with respiration. Additionally, all apparatus must be clean to avoid contamination that could compromise the results. Finally, temperatures should not exceed levels that can destroy yeast cells completely, specifically above 60 degrees Celsius. Following these precautions helps ensure that the investigation is both safe and scientifically valid. Now, let's talk about the principle behind this practical. Yeast respires aerobically, which means it breaks down glucose in the presence of oxygen to release energy. To observe this process, we use methylene blue, which is a redox indicator. When the methylene blue is blue, it's in its oxidized form, meaning there is no respiration happening. When it becomes colorless, it's in its reduced form, which shows that active respiration is taking place. Essentially, the speed at which the color changes tells us about the rate of respiration. The faster the color fades, the higher the rate of aerobic respiration in the yeast. Then, the objective of this practical is to investigate how temperature affects the rate of aerobic respiration in yeast, using methylene blue as an indicator. As a reminder, respiration is the process by which cells release energy from glucose. In yeast, respiration can occur aerobically, with oxygen, or anaerobically, without oxygen. This experiment focuses on aerobic respiration, which is more energy efficient and produces carbon dioxide and water. The rate of respiration can be influenced by environmental factors, with temperature being particularly important. To study this, we can use various experimental methods. One common and effective approach is to use a colored indicator like methylene blue, which is non-toxic to yeast and allows us to observe and measure the rate of aerobic respiration. Next, it's important to be familiar with the apparatus and materials needed to carry out this experiment. Apparatus required for investigating the effect of temperature on yeast respiration includes a yeast suspension, glucose solution, test tubes with a rack, methylene blue dye, a stopwatch, and a temperature-controlled water bath. Materials and chemicals needed are 1 to 2% glucose solution, 0.1% methylene blue solution, and optionally a buffer solution to maintain the pH around 7. Now, let's look at the independent and dependent variables in this experiment. The independent variable is the factor that the experimenter deliberately changes. In this investigation, it is temperature, as we are examining how different temperatures affect the rate of respiration in yeast. These temperatures are maintained using water baths set to specific values. The dependent variable is the factor that is measured or observed during the experiment. Here, it is the rate of respiration in yeast, which we measure by recording the time taken for methylene blue to change from blue to colorless. A shorter time indicates a faster rate of respiration. It's also important to consider controlling other variables to ensure a fair and reliable experiment. When we study the effect of one factor, all other variables should be kept constant. All factors other than the independent variable should be kept constant. For example, the volume or concentration of methylene blue must be the same in all test tubes, as adding more dye can make the color take longer to disappear. Similarly, the volume or concentration of yeast suspension should be consistent because a higher number of yeast cells increases the rate of respiration, causing the dye to lose color more quickly. The concentration of glucose must also be controlled, since limited glucose reduces respiration by providing less substrate for energy release. Finally, the pH level needs to be maintained, as the enzymes involved in respiration are sensitive to pH changes. Using a buffer solution helps keep the pH constant and prevents enzyme denaturation. Now, let's go through the procedure for this experiment. So, first, we prepare a yeast suspension in glucose solution because glucose acts as the food source for the yeast, allowing it to carry out respiration. Adding a few drops of methylene blue serves as an indicator to monitor respiration. It starts blue and becomes colorless as the yeast respires. Next, the test tubes are placed in water baths set at different temperatures. This is done to investigate how temperature affects the rate of respiration. As soon as the tubes are placed in the water baths, a stopwatch is started to measure how long it takes for the blue dye to become colorless. Observing the time taken for the color change gives us a direct measure of the yeast's respiration rate. A shorter time indicates faster respiration, while a longer time indicates slower respiration. By recording the time for each temperature, we can compare how different temperatures influence the speed of aerobic respiration in yeast, helping us understand the effect of temperature on enzyme activity and cellular processes. After carrying out the procedure, we can observe how temperature affects the rate of respiration in yeast by looking at the time it takes for methylene blue to change from blue to colorless. At 10 degrees Celsius, the color change takes the longest, around 120 seconds, indicating that respiration is slow at low temperatures. As the temperature increases to 20 degrees Celsius and 30 degrees Celsius, the time decreases to 90 seconds and 60 seconds respectively, showing that the rate of respiration is increasing. The fastest rate is observed at 40 degrees Celsius, where the color changes in just 30 seconds, suggesting that this is close to the optimum temperature for yeast respiration. However, at 50 degrees Celsius, the time increases again to 80 seconds, indicating that higher temperatures start to slow down respiration, likely because the yeast cells or enzymes are being damaged. This pattern clearly demonstrates how temperature influences the rate of aerobic respiration in yeast. Next, we calculate the rate of respiration using the formula, which is 1 divided by the time taken for the color change. When we apply this, we see that at 10 degrees Celsius, the rate of respiration is very slow. As the temperature rises to 20 degrees Celsius, the rate increases slightly, and at 30 degrees Celsius, it rises further. The fastest respiration occurs at 40 degrees Celsius, showing this is close to the optimum temperature for yeast activity. However, when the temperature is increased to 50 degrees Celsius, the rate drops again, indicating that higher temperatures begin to inhibit yeast respiration. This shows clearly that temperature has a significant effect on the rate of aerobic respiration in yeast. Then, we use the calculated rates of respiration to plot the graph. Each rate, obtained by dividing 1 by the time taken for methylene blue to lose its color at a specific temperature, becomes a point on the vertical y-axis, while the corresponding temperature is plotted on the horizontal x-axis. By connecting these points, the graph clearly shows the trend. The rate of respiration increases with temperature up to 40 degrees Celsius, then decreases at higher temperatures due to enzyme denaturation. In other words, the numerical results we calculated directly provide the data points that form the curve, visually illustrating how temperature affects yeast respiration. So, by looking at the graph, we can see that the rate of respiration increases steadily as the temperature rises from 10 degrees Celsius to 40 degrees Celsius. The highest rate occurs at 40 degrees Celsius, which tells us that this is close to the optimum temperature for yeast respiration, where the enzymes are working most efficiently. However, when the temperature goes above 40 degrees Celsius, such as at 50 degrees Celsius, the rate drops sharply. This decline happens because the high temperature causes enzyme denaturation, damaging the enzymes involved in respiration and reducing the yeast's ability to respire. So overall, the graph clearly demonstrates how temperature affects the rate of aerobic respiration in yeast. So, the experiment shows that as the temperature increases up to around 40 degrees Celsius, the rate of respiration increases, and the methylene blue solution turns colorless more quickly. The trend refers to the general pattern or direction that the data shows, in this case, how the rate of yeast respiration changes with temperature. So, by looking at the results, the trend shows that as the temperature increases up to around 40 degrees Celsius, the rate of respiration increases, and the methylene blue solution turns colorless more quickly. This happens because higher temperatures give molecules more kinetic energy, leading to more frequent and effective collisions between enzymes and substrates, which speeds up the reaction. However, when the temperature rises above 40 degrees Celsius, the rate of respiration decreases, and it takes longer for the color change to occur. At these higher temperatures, the enzymes begin to denature, meaning their active sites lose their specific shape, preventing them from forming enzyme-substrate complexes and reducing the yeast's ability to respire efficiently. So, the conclusion from this investigation is that temperature has a significant effect on the rate of aerobic respiration in yeast. The optimum temperature is around 37 to 40 degrees Celsius, where respiration occurs at its fastest. At high temperatures above 40 degrees Celsius, enzymes begin to denature, which slows down respiration, while at low temperatures below 20 degrees Celsius, molecular movement is reduced, leading to a slower rate of respiration. Additionally, methylene blue serves as a simple and effective visual indicator to monitor the rate of yeast respiration. And that wraps up Unit 10.1 on respiration. I hope you found it helpful. We'll be back soon with the next topic, so make sure to stay tuned.